And we're gonna start by looking at the warm-ups. And yeah, I mean, obviously we're starting to get more into lots of new material now, right? I mean, the first couple of weeks we're just kind of reviewing the chemistry, but from this point on, we're gonna be just adding in more and more new concepts. So it's gonna be more important than ever to be keeping up. This is definitely not the kind of class where you can just cram right before your exam and just pull a rabbit out of a hat. You know, usually if you try to cram right before the exam, you'll not do so well. So these warm-ups are a way to, again, kind of check in, are you keeping up here? Let me go to, all right, la, 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 la. Hold on. Module. I need to share my screen here. Share. And we're going to warm up and quiz statistics. All right, just again, as a reality check, so you can have a sense of where you are compared to other people. You know, quite a few people on this warm-up got less than half of them. You know, if you are one of those people, you know, that's probably an indication that you probably want to put more time, you know, outside of class into kind of studying and getting up to speed on stuff so you can keep up. Like I said, like kind of leaving all your studying for the end will probably not be a good idea. In fact, it won't be a good idea. All right, choose all that are true about cilia. And again, here, this was pretty good. Most people got this. You know, they are motile, they move. They're the dudes that will, you know, sweep mucus across the respiratory epithelium, or will, again, sweep the egg towards the uterus. They have a system of microtubules and little dynein proteins that physically make them move back and forth. So microtubules, and they move. You know, this one, exogenous ligand that binds and blocks a receptor. You know, again, an antagonist. What if it binds and it actually activates the receptor?
Agonist.
That's an agonist, exactly. So, and again, there's a lot of drugs, both pharmaceuticals and recreational, that act either as agonists or antagonists, will bind the receptor and either activate it or block it. Which of the following cell-to-cell attachments electrically connects adjacent cells? Again, most people here got it, gap junctions. They actually connect the cytoplasm so ions can freely pass. We're gonna see these connecting like smooth muscle cells, cardiac muscle cells, certain kinds of electrical synapses and neurons. So gap junctions. Yeah, conjunction junctions are Schoolhouse Rock, tuxedo junctions are Glenn Miller. What do desmosomes do?
They keep the cells and neighboring cells like anchored together.
Exactly. They're the ones that provide that structural integrity so you can't pull the cells apart even under stress. And tight junctions. What about tight junctions?
They're like a Ziploc that seal the cells together so that things don't come through.
Yeah, exactly. So I mentioned those specifically in the case of the transepithelial transport, where you want to make sure that molecules aren't cheating by wiggling between the cells to get from one side of the sheet to the other. You need to make sure the cells are all connected with tight junctions. Again, it's best way, kind of a good simile is they're like kind of like the Ziploc, where the two things, it's still in a Ziploc bag, the two sides of the bag are still separate pieces of plastic, but they interlock in such a way that they make this seal that you can actually turn the bag upside down and no water comes out, right? So it's not like they're, so it's still two sides of a bag, but they're so tightly kind of folded into each other that it makes this passage-proof seal. Drugs like caffeine, morphine, LSD, THC, beta blockers, albuterol, lots of different ones. They act as exogenous ligands. And again, here, a lot of people they bind the receptors on the cell membranes and affect cell behavior. And we're going to look at this in more detail today, this whole idea of what is binding, how does that binding event actually start the whole signal transduction pathway. This disrupts cell membranes. They don't. There are things that do actually get into the membranes. Things like some of the anesthetics actually work, they think, by getting into the cell membranes and altering the behavior of things that are actually other membrane proteins. Early on, like when I first started teaching this class, people thought maybe, nobody knew exactly how people got stoned. They knew THC was the active ingredient in marijuana, but nobody knew why it worked. And THC is lipophilic. So one of the theories was maybe THC gets into the cell membrane and affects the behavior of other channels and stuff. But eventually, it was actually, I forgot the dude's name, some dude in Israel found the receptor that THC actually binds to, and they realized, oh, it's actually binding a cell receptor. And actually, once they realized that they found this receptor, which they gave it a hilarious name, they called it the Anandamide Receptor, because Ananda is Sanskrit for bliss. So the receptor on the neurons that THC was binding to, they called Anandamide receptors. And then they figured there's got to actually be an endogenous ligand, some native neurotransmitter that's using that receptor. It doesn't make sense that the receptor exists if there's nothing naturally occurring in the body that's using it. And so then they looked, and sure enough, they found, the endogenous cannabinoids. So in the main neurotransmitter that binds these receptors, they called anandamide, again, after Sanskrit for bliss, which is so, but yeah, the reason they found this particular neurotransmitter, or the reason they were even looking for it, was first they had discovered the receptor that THC binds to, and then decided there's got to be an endogenous ligand that also uses that. Let's see. Which of the following dramatically increases the cell surface area? That's the microvilli. Again, cilia are not. Cilia are, they're going to increase surface area, obviously, because they are making it up and down, but that's why I was careful in the wording, is primarily for dramatically increasing cell surface area. Cilia are primarily about sweeping things along the surface of the cell. Microvilli don't move. They're not motile, and their kind of raison d'etre in the universe on a cell is to just increase surface area. So, are there any questions about any of these?
For the last question, question 5, you made it, at least on my quiz, you could mark multiple. So I put both cilia and microvilli, but it marked me wrong.
All right. You know what, then I will, it's still not exactly right, but I can give you a little bit of credit on that. I didn't mean it to be a multiple answer correct. So I can go back and fix that.
Okay, thank you.
Did you mean for, because like a lot of mine were multiple choice, so.
Usually I say in particular, like choose all that are true. You know, so if there are more than one possible answers, it doesn't mean there is more than one answer, but if there's more than one possible answer, I usually, or almost, at least unless I spaz, I tell you that there is more than one potential correct answer. If I don't say that, then you should assume like choose the best answer. And there's something in the way that Canvas presents the question even. It's like, I think the little boxes you can check look different if there are more than one possible correct answers versus one correct answer. So, but yeah, in general, I will say something like choose all that are true or something like that. If I am kind of leaving open the door for more than one correct answer there. Any others?
I have a question about the first one. I was not sure because I just put one. Can you explain a little bit about cilia? Like what is true about cilia?
Yeah, cilia, remember we talked about, they're the ones that are on the top of the cell and are kind of moving. They're motile, they move. So they're like sweeping things along across the cell. And also we talked about the microtubule structure within them, the 9+2 structure microtubules, where those microtubules are getting pulled next to each other. That's actually causing the bending and waving of the cilia. So those were the two things that were true there.
Okay, thank you.
Okie dokie. So we are going to continue talking about, in fact, we haven't totally finished membrane proteins even yet, but we're almost done there. And then we're gonna continue talking about lots more things about cells. And so let's do that. Stopping the share. Okay. All right, so we've been talking a bunch about membrane proteins. And again, there is so much important stuff that happens at the membrane of a cell. That's that boundary between, you know, the living thing and the rest of the universe. And that's where these things are going to be interacting with other things around them and trying to figure out what's happening or communicating with each other. So it's important that we have a good sense of this boundary and all the different things that are happening at this boundary. Again, if we wanted to just briefly review, what are some of the functions of these membrane proteins?
Transportation.
Yeah, there's transport. We talked about getting things in and out through either channels or carriers or more complicated things like that act as pumps and symports and antiports and all that. And then we also go on.
Cell ID. I was about to say another function. Sorry.
Yeah, cell identification. We're going to talk about this more when we get in the immune system, but the idea that each of your cells basically has a little barcode on it that says this belongs to your particular body, and your immune system can see, does this cell belong here or is it here and we need to deal with it. What's some more?
Cell to cell attachment.
Cell to cell attachment. Right. Whoop, I just misspelled that. Oh, yeah. I'm realizing, I think because I like get up really early and by the time we're in the middle of this, I'm getting slightly fuzzy. I have like typos in some of my lectures. So cell-to-cell attachment. We talked about desmosomes, tight junctions, gap junctions. You know, there's some other ones as well, but that's the main thing there. What are some more functions of membrane?
Receptors.
Cytoskeletal attachment.
Yeah. Cytoskeletal attachment points. But wait, there's more.
Receptors.
Yeah, receptors. And we talked a bunch. We're going to talk much more about receptors today. Anything else?
Endoexocytosis with clathrin
Yeah, we
protein.
talked about clathrin protein that was helpful for in those coated pits, where parts of the membrane that are specifically for endo and exocytosis. And again, as I mentioned, that actually kind of makes a cage around the vesicle once it's inside and stabilizes it. So it kind of holds together rather than just being like a little bubble that can fall apart. And then another big one that we haven't talked about that I'll add into the list, enzymes. So the idea of having a membrane protein that's an enzyme, this will make sense if there is some reaction that you want to catalyze right near the membrane, right? If there is some reaction that you want to happen at the membrane, then it makes sense to anchor that enzyme at the membrane. For example, when we get to the digestive tract, the enzymes that, you know, if this is an intestinal epithelial cell, here are, you know, the stuff I'm digesting. Let's say these are disaccharides. The enzymes that do the final breakdown are actually anchored on the membrane of the intestinal epithelium. We call them the brush border enzymes. Because when we finally do the final breakdown and we have an individual glucose, we want that individual glucose to come into existence right near the epithelium, because that's where we're going to bring it in to absorb it. Right? So when we talk about digestion much later in the semester, you're going to have kind of the enzymes for the more coarse breakdown of the food, like that are coming in from the pancreas and things like that. Those are going to take, you know, starch and cut it into shorter chains of carbohydrates, or take proteins and chop it into shorter peptides and stuff. But the enzymes that do the very final fine-grained breakdown into individual monosaccharides or individual amino acids, those are actually anchored on the membrane of the cells, where you are now going to just absorb those nutrients. Right? So that, does that make sense? You know, you want the reaction and the products to show up right near the membrane, so you should have the enzymes at the membrane. When we talk about signal transduction in just a little later today, there are important things that are part of the message from a receptor binding to something happening in the cell that has to happen near the cell membrane. And we're going to see some important enzymes that are part of signal transduction that are anchored in the cell membrane. So there's going to be a number of examples of enzymes that are anchored in cell membranes. So that's another kind of membrane protein we'll see. Okay. So now I'm going to talk about signal transduction. So signal transduction. This is the idea that you have a receptor. We talked about a receptor binding some ligand, and then something happens. It depends on what the signal was. It might, if it's some neurotransmitter, the neurotransmitter binds a neuron and it's going to create some electrical activity in the target cell. Or maybe it's a hormone. It might be insulin binds some cell and it's going to like turn on some things to tell the cell to take up glucose. Or who knows, there's lots of different things that might happen as a result of some ligand, some signal coming in and binding a receptor. So signal transduction is the idea that we're taking some signal, some ligand binding some receptor, and we're translating it into some actual results, some action in the cell. The transduction is kind of like translation. So we talked, so for instance, we talked about ligand gated channels. So the idea that you have a channel that will be closed, but it will open if it binds a particular ligand. So there are two major different ways that this can happen. We can have directly gated. So in directly gated channels, so an important one of these, the nicotinic cholinergic receptor, which we're going to look at in much more detail later. Like, for instance, e.g., nicotinic cholinergic receptor. Don't worry too much. We're going to talk about what nicotinic and cholinergic and everything means later. For right now, I just want to throw out that name so when it comes back later, this will all be clear. But basically, you have your cell membrane. You've got the receptor. And it binds acetylcholine. ACH just means acetylcholine. Acetylcholine is going to be one of the important neurotransmitters that we spend time looking at. So here we have my little acetylcholine molecule. This is my ligand. This is going to be, here is my receptor. And again, there's this perfect fit where the acetylcholine binds my acetylcholine receptor. And when that binding happens, it automatically opens up a channel that lets sodium in. What am I doing there? So this is one piece. It's this mechanical thing. When acetylcholine binds, this channel opens. If acetylcholine is not there, the channel is closed. So that's why it's called directly gated. There's no middleman here. So directly gated, acetylcholine binds this receptor. When that binding happens, there's an actual conformational change of this membrane protein that opens up the channel and sodium can rush in. When acetylcholine is not docked in its binding site, then the channel's closed. So does that make sense? It'd be like you push the button when, as long as you're pushing the button, it's pushing on a spring that opens up the door. But as soon as you let go of the button, the door slams shut again. So this is a directly gated channel. There are very important directly gated channels. You know, this nicotinic cholinergic one is one of them. It also means that there is no wiggle room for what happens. It's like acetylcholine is bound, it opens and lets sodium in. Acetylcholine is not there, it's closed. So there's no options. It's just either open if acetylcholine is bound or closed if acetylcholine is not bound. And then I'm going to say versus indirectly gated channels. And these use second messenger. So second messengers and G protein coupled receptors. So that's what we're going to talk about right now. What the heck am I talking about when I say G protein coupled receptors and second messengers? I've already kind of teased the idea of second messengers when we were talking about the nucleotides. Remember, like cyclic AMP, cyclic GMP were examples of second messengers. And now we're going to finally see how they actually fit into this whole story. And it's going to look a little, and I should say, okay, so I'm going to go to a new page here. So what I'm going to talk about now is signal transduction using G protein coupled receptors and second messengers. So the basic idea is signal transduction means there's, oh, I keep changing the color. The signal is going to be the original ligand coming in and binding the receptor. And signal transduction is there's some actual thing that happens in the cell as a result of that. In the examples I was just giving, you know, the result is we open a channel, for instance. But in other cases, the result of binding a receptor might be turning on some enzymes to make reactions happen in the cell. Or the response might be turning on protein synthesis and making stuff. There's lots of different outcomes that can happen in the cell as a result of a receptor being bound by its appropriate ligand. So what we're talking about now is more general than just using receptors to open or close channels. This is going to be one of the things you can do is open or close a channel like I was talking about here, but we can also use this second messenger system to turn enzymes on or off, like I said, or, you know, turn on or off protein synthesis or do different things in the cell. So it's going to be a much more flexible kind of system. It's not here, acetylcholine binds this receptor and the only thing that happens is you open up a sodium channel. It's just a self-contained one-trick pony here. These second messenger systems are going to be a lot more flexible, but as a result, they're also going to be a lot more complicated. So let's dig in. All right, so I'm going to first draw my cell membrane. And then obviously, we're going to need a receptor. And a receptor that binds a particular ligand. So this is my receptor. So this so far should look kind of familiar. A ligand binds a receptor. But now what we're going to do is add in a whole other bunch of parts and pieces that are actually going to be necessary for that binding event to actually result in some action inside the cell. So like I said, these are G protein coupled receptors. So the receptor has these three little subparts on there. So these are the actual G proteins. The G protein, it's actually got three parts. The main part that's gonna like be, do a lot of the action is the alpha subunit, but it doesn't matter so much. You can just think of it as the G protein. So when the membrane protein, this receptor is just sitting here and has not bound a ligand, this G protein is kind of waiting to go and is inactive. It's kind of bound onto the receptor, and it's called a G protein because it binds GTP, guanine triphosphate. So right at this point, it has, I think, GDP on it if it's not active. But if it's going to activate, it's going to bind GTP and become active. So we have the first thing that's going to happen is the ligand is going to bind the receptor. And when that happens, that's going to activate the G protein. And when the G protein activates, it actually lets go and starts drifting through the membrane. So this is now, maybe I'll make it like, I'll put like, this is like activated. So this is kind of part 2 now. So part 1, the ligand bound the receptor. That actually, and we're going to talk about exactly how this works, but it actually causes a conformational change in the receptor. When the ligand binds the receptor tweaks its shape a bit. That tweaking the shape of the receptor is actually going to catalyze the activation of the G protein. So now the G protein here is moving through the membrane. and where it can do things, it can activate things. There are many, many different examples of different kinds of second messenger pathways. I'm going to describe one, common type of second messenger pathway right now. It's not the only one. So after I talk about this one particular pathway in more detail, I'll then step back and tell you about other variations on a theme. But we'll start with one particular kind because it's illustrative of how these second messenger pathways work. So this first step, again, binding between the ligand and the receptor. Second step, activating the G protein, which is now on the move through the membrane. Again, one of the classic things it does is the G protein can slide over and activate another membrane protein. This one here, I'm going to give it a name. It's going to be a bit of a mouthful. Adenylyl cyclase. I can probably, since this is a cell membrane here, I'm going to erase that to give myself a little more room. Adenylyl cyclase. This is an enzyme that catalyzes production of cyclic AMP. And we'll see that in just a few moments. All right, so this G protein, this activated G protein drifts around and all of a sudden comes over to here. And again, this is my active G protein, my activated G protein. It's going to activate the adenylyl cyclase. So that's going to be part 3. We activate adenylyl cyclase, which is then going to start producing cyclic AMP, which is going to start building up in the cell. So we can say this is kind of part 4. Right? Ligand bound the receptor, that activated the G protein. The G protein came over, activated the adenylyl cyclase. The adenylyl cyclase is now making lots of cyclic AMP. And remember, I called cyclic AMP a second messenger. The reason it gets that name is you can think of this ligand here as being the first messenger. That was the original messenger that came in that the cell recognized. But now, after all of this activity we've just been seeing, we now have a signaling molecule showing up on the other side of the membrane inside the cell. This is my second messenger. So, and again, this is going to be for ligands that are not lipophilic. If I have ligands that are lipophilic, things like a steroid hormone, they don't need this whole system. They can just drift through the membrane and get into the cell where they can do stuff, right? So a steroid hormone is actually going to drift through the outer membrane. It's actually going to drift through the nuclear envelope and start binding and activating things within the nucleus of a cell. The ligands that we're talking about here are the ones that are non-penetrating. they can't get in the cell, so they bind a receptor on the outside of the cell, and then through this process, they are now creating the second messenger inside the cell. So now there is a signal inside the cell that can continue this process.
Could you explain what lipophilic means real quick?
Lipophilic, it means hydrophobic. It means it likes lipids. So it's something that's nonpolar. Yeah, lipophilic and hydrophobic and nonpolar are all kind of the same thing. Yeah, I think it's kind of the messaging. Like you could say hydrophobic, which sounds kind of more pejorative. Oh, you're scared of water. Or lipophilic, they love fats. Makes it sound a little more positive. So, so cyclic AMP, but cyclic AMP is just another messenger. We still gotta use it to do something. Often what happens next is the cyclic AMP activates a protein kinase A, which is, so let's see. So what does a kinase do?
It's phosphorylation.
It phosphorylates things, right? So this protein kinase A, this is going to phosphorylate things. Phosphorylates proteins. So protein kinase A phosphorylates proteins. So this is now going to go off and it's going to phosphorylate something. One kind of protein it can phosphorylate is a channel protein. And so that might end up opening or closing a channel. Another kind of protein it can phosphorylate can be an enzyme. And it could be that phosphorylating the enzyme activates or inactivates the enzyme. Or lots of other stuff. So this idea here, so now if we're continuing with our like numbering system here, ligand bound the receptor on the outside of the cell. That was where it all started. The receptor binding the ligand activated the G protein. The activated G protein, that was number 2. Number 3, the activated G protein turned on adenylyl cyclase, this membrane protein here, which makes cyclic AMP. So kind of the number 4 of this was cyclic AMP starts building up in the cell. Number 5 would be that cyclic AMP activates the protein kinase. You know, number 6 is that protein kinase phosphorylates some protein. Maybe it's a channel protein and it opens or closes a channel. Or maybe it's an enzyme protein and it's turning on or off some enzyme. It's creating some action, some activity in the cell. So this is a classic second messenger pathway that I've just drawn here. Gesundheit. Are there questions about what I've drawn right here?
I have a question. So second messengers like cyclic AMP, can they be activated by other things like other than G-coupled proteins, or are these always things we'll see together?
There are other second messenger pathways that don't use cyclic AMP, but cyclic AMP is always, as far as I know, is always dealing with G proteins.
Okay, so we'll always kind of see, or mostly see them together, the G-coupled proteins activating cyclic AMP.
Yeah, but sometimes instead of cyclic AMP, you'll make cyclic GMP.
Okay.
Or in other ones, the G protein, instead of activating adenylyl cyclase, activates another enzyme in the membrane that ultimately releases calcium in the cell, and calcium then acts as a second messenger. So
okay, I see the
what shows
difference.
up, this G protein, step 2, up to step 2, that's always pretty much going to be there. But once you get past step 3, we're already getting to a very specific pathway.
Okay, that makes sense. Thank you.
Yeah. Yeah, so like I'll talk about like the calcium pathway. You have phospholipase C. Instead of adenylyl cyclase, there's a really common pathway using an enzyme phospholipase C as the third step, which results in ultimately a bunch of calcium in the cell. And then that's acting as a second messenger instead of cyclic AMP. And we'll talk about why this is important in a few moments. While I have this big messy picture up here, there's another important point we have to make. A few important points we have to make. So we'll come back to this. Let's talk about why go through such a big, complicated process. What are the benefits of doing something like this?
Like the flexibility that you were talking about versus it being just one thing, like gated. So
right. So a lot of, do you see the flexibility? I could have this ligand. Let's say this ligand is insulin. You know, what happens in the cell totally depends on the rest of the second messenger pathway. You know, and it's going to be different in different kinds of cells. If it's a liver cell, this is ultimately going to turn on enzymes that break apart glycogen and, or actually turn on, if you have insulin, you're making glycogen. You know, so it's going to turn on enzymes that catalyze the production of glycogen inside the liver cell. This is some other kind of cell, it's just going to turn on other things, telling them to, you know, transport glucose into the cell. You know, depending on what the target cell is, what you want to do when you've got insulin as the message might be different. So we can have the same ligand, but have different outcomes within the cell. So maybe I can, you know, so this is our examples, what Sari said as a broad idea of flexibility. Right, we can have right? So the same ligand can result in different outcome in the cell depending on what second messenger pathway you've hooked up. The other thing though, wait, where is this, there it is. Another thing is, let's say I have a particular outcome, like releasing glucose into the bloodstream. So I want to like turn on an enzyme that breaks down stored glycogen to release glucose. It turns out that there's lots of different things that might tell a liver cell that we want to up the amount of sugar in the bloodstream. One thing would be the ligand, the signaling molecule, which is glucagon. It's a hormone that tells the liver we need to release stored carbohydrate. But other things that might release glucose would be like these glucocorticoids, like these hormones that are part of your stress response. Or other things might be thyroxine, thyroid hormone, which is kind of increasing your metabolic rate and you're going to need more sugar. So there might be lots of different signals, initial signals, first messengers, that ultimately you want to result in the exact same outcome. You want to turn on a enzyme that breaks apart glycogen and releases glucose into the body. So you can have the ligand and the receptor be different, but then ultimately turn on the same pathway in the cell. So they can all kind of converge on the same result, right?
So is it like amplifying their signals?
So that we're going to get to that. That's going to be another thing. Well, let's hold off on that just for a moment. But that is going to be an important thing going on here. But if we want to continue with this idea of flexibility, we can have different ligands can all result.
Professor, I think you're on a different.
What's that?
Never mind, it fixed itself. It was on a different screen before.
Yeah, no, there's some, for whatever reason, there's some lag here. I'm noticing it can result. So here, the idea that you have many different ligands that might all ultimately turn on some enzyme to release glucose into the cell. So these are both examples of flexibility. You can have, adrenaline in some cell is going to dilate muscles, and other cells it might contract muscles. In other cells, it's going to speed up your heart. In other cells, it's going to dilate your pupils. Depending on where the receptor is, the outcome's going to be really different. You have a different pathway to have a different outcome. So these are all examples of how you get much more flexibility by using second messenger pathways. And then we can get to what Nikki was just talking about. So this idea of, we have one initial moment where the ligand binds the receptor here, and it activates the G proteins, but then the G proteins start turning on adenylyl cyclases. Adenylyl cyclases start making lots of cyclic AMP. Cyclic AMPs are turning on these protein kinases, and those, once they're active, start phosphorylating lots of proteins. So at each stage, we have this multiplication going on. So there is this huge amount of amplification that happens. It's this big exponential increase. Like, in fact, I have some little factoid here. What is it? I wrote it down. Here it says, one molecule of epinephrine. So a liver cell gets a single molecule of adrenaline, epinephrine, it can mobilize the release of 100 million molecules of glucose into the bloodstream. Right? So there's this huge amplification that happens where one binding event starts creating lots of second messengers, which start activating lots of protein kinases, which start activating lots of enzymes. So you all of a sudden have this huge response. Does that make sense? Right, so if we go back to our list of the benefits of second messenger pathways, we had flexibility as the first benefit. Second benefit is going to be we have amplification. Somebody here. So that makes sense. And then the third one is going to be, I'll kind of write out here, kind of lots of opportunity for control. I'm going to come back to this in a moment. I want to talk, this will make more sense when I give you a little more information. So let's come back to this big messy picture here inside the cell. I need to make a little room for myself, so I'm going to erase some of this stuff which you already know. Okay. So imagine my first messenger comes in. It activates the receptor, turns on the G protein, turns on adenylyl cyclase, makes lots of cyclic AMP, starts doing whatever it's going to do. I need this to end at some point, right? If this ligand only binds for a little while and then it goes off and does its thing. Remember, we talked about binding is temporary. It binds, it lets go, it binds, it lets go. Depending on the affinity, it might hold on longer or less long. But at some point, the ligand is cleared out of your system and it's gone. So the message is done, at least from the standpoint of the first messenger. But as long as there's cyclic AMP in the cell, the message still keeps going, right? So we need to make sure we get rid of this cyclic AMP or the message will never stop. So we need something that breaks down that cyclic AMP to actually turn off the signal, turn off the message. So we have a particular enzyme that breaks down the cyclic AMP, and it's called phosphodiesterase. Usually abbreviate it PDE. So this is going to be important. So basically the phosphodiesterase will come in and start breaking down the cyclic AMP, which then is going to turn off the rest of the thing. So this is important. If we didn't have this, the signal would never stop. So this eventually stops the signal. It stops transduction. So does that make sense? One of the most like kind of popular, profitable drugs out there is a phosphodiesterase inhibitor. So Viagra, sildenafil, the stuff that guys will take to help with erections, for erectile dysfunction. Viagra is a phosphodiesterase inhibitor. So, okay, you know. So how does this help a guy with erections? It turns out that erections ultimately result from dilation of blood vessels that help more blood come into the penis and engorge the spongy tissue and all that. It's all mediated by a second messenger pathway. In this case, using cyclic GMP instead of cyclic AMP. But if we go back to our picture here. So imagine we have this pathway and, 6, the result of it is an erection. But phosphodiesterase is always in there. So you turn this on and you have an erection, but in the background, you've got phosphodiesterase, stopping the pathway. So the guy's getting soft again. So that's kind of how it normally works in somebody who has Viagra. This phosphodiesterase is inhibited. So if you turn this pathway on, it stays on. It doesn't turn off, right? Because you don't have the enzyme that's normally there that is turning off the signal. So does this mean, so if a guy just takes Viagra, will it give him an erection? No, it doesn't. It doesn't turn on anything. It doesn't actually activate the pathway that creates an erection. But what will it do?
It will make it last longer.
Exactly. Once it gets turned on, it will make it last longer and keep the signal going strong rather than have it back off due to the second messenger getting broken down. So this should make sense. This is not just like kind of a little titillating side note. This is kind of at the core of kind of understanding these second messenger pathways here. We're going to meet these again and again and again. We're going to meet phosphodiesterase. Phosphodiesterase is at the core of understanding how the rods and cones turn your light into electrical signals in your retina. You need to know phosphodiesterase, throughout the semester. You need to know all of these G proteins throughout the semester. This is really an important, and this is also where, again, a lot of we talked about a lot of drugs that work as being agonists or antagonists, where they actually are interfacing at the level of the receptor. But a lot of other drugs work further down the pathways, like Viagra. Viagra doesn't work at the receptor. It works inside the middle of the second messenger pathway here. So that's another important place you can go in and affect the function of signaling in the cells. Second messenger pathways. So I just described one that uses cyclic AMP. Again, a lot of other ones use cyclic GMP as their second messenger. And those are kind of more, they're pretty similar to the one I just talked about. Cyclic GMP, the one, again, involved with erection, the ones involved in phototransduction, use cyclic GMP instead of cyclic AMP. There's another very different but important type of second messenger, and that's calcium. So in some second messenger pathways, the G protein, instead of activating adenylyl cyclase, will activate this thing called phospholipase C, another enzyme in the membrane. But that is gonna, I'm not gonna go into the details. It's basically gonna do a chain reaction of things that ultimately increases calcium in the cell. This is another type of second messenger. The calcium binds this, what is it, calmodulin, this other protein that instead of, we end up having another another kinase that ultimately gets turned on. It's like protein kinase, what is it? I think it's C instead of A. But so you ultimately turn on protein kinases and stuff, but it's through a different pathway. Instead of using cyclic AMP or cyclic GMP as a second messenger, you can use calcium. These can all be second messengers. And you can have different second messenger pathways that are happening at the same time. And you can have second messenger pathways that mess with each other. So, coming back over to here, I might have this one second messenger pathway mediated by cyclic AMP, and whether it's on or off depends on the amount of phosphodiesterase. I might be turning phosphodiesterase on or off with some calcium mediated second messenger pathway from some other ligand. So I can actually have different second messenger pathways that affect the behavior of each other and modulate each other's behavior. So that's when I talked about lots of opportunity for complex control. I can have different second messenger pathways that actually affect the behavior of each other. So it's not like, it's not a linear system. It's not like they are all independent. They actually affect each other. And when one's running or not, it might actually determine whether another one actually works at all or how it works. So it gets kind of mind-bogglingly complex when you start looking at all the possibilities for the signaling in the cell. And then, so I'll mention this. This is an aside. Please don't, for the stuff you're responsible for your tests and stuff, you should understand all the stuff I've been talking about here, which is a lot. That being said, when a ligand binds some receptor, there might be more than one second messenger pathway that gets activated at the same time from this one receptor. It can actually get more complicated than just one second messenger pathway. And even weirder than that, like the people that have been studying exogenous ligands, the ones I'm more familiar with are the psychedelic drugs. You can actually have, even though most psychedelics all attach on this particular serotonin 2A receptor, exactly what happens on the other side actually can be different, slightly different, depending on what kind of drug. Just because you have a ligand that binds the receptor doesn't necessarily mean it's the exact same effect on the cell side. So the reality is that the real story it's a rabbit hole. It's like, whoa, down the rabbit hole. When you start really, most of the stuff in the cell, Like it's easy to tell a kind of more superficial story about what's going on that's basically true. But once you get into the real details and look closer and closer and closer, things usually just get weirder and weirder and more complicated. And you kind of hit a wall of like, I don't know what's going on here. So, but again, this, don't worry about that. Where are we in terms of time? So right here, let's come back here just for a moment. I was talking about the idea, it all starts when the ligand binds the receptor. And again, what kind of a molecule is a receptor?
Protein.
A protein.
It's a protein. And when the ligand binds the receptor, there's some kind of a little conformational change, some little movement of the receptor that actually changes the shape of the other side, of the cytoplasmic side of this receptor, which is going to be what catalyzes the activation of the G protein. So what I'm going to do now is give you an example of how that can happen. And what does it actually mean to bind, for a ligand to bind a receptor? This example goes into more detail than you need to know, but the basics you should know. So, it also reviews a lot of the chemistry concepts, which are actually important for you to make sure that as you're seeing them in context, you can say like, oh yeah. So what we're going to do now is go on a little tour of a serotonin molecule binding a serotonin receptor. And what the heck does that actually mean? We could just say magic. Serotonin binds a receptor and that turns on something. It activates a G protein. It rolls off the tongue and then you think, oh, I understand. It's like, but what does it mean to bind a receptor? How the heck does binding, whatever that means, actually activate a G protein? So that's what we're going to do now. So we're going to go, hold on, Physio, PowerPoint. Okay, I'm going to share. Okay. All right, so this is a I did not make this PowerPoint presentation. This was made by a friend who is, or was a scientist at Pfizer designing antipsychotic drugs. And part of designing drugs that work, in the neurons is understanding like how do the normal receptors or how do the normal ligands bind the receptors and what kind of shaped molecules could we make that are going to fit in there and what kind of effects are they going to have? So they have a lot of motivation to really understand how a ligand binds a receptor. This presentation is particularly about serotonin, which is one of the really common neurotransmitters in the brain. Most people have heard of serotonin. It also plays lots of other roles. We're going to meet serotonin as a major player in kind of hemostasis. Like, when, in fact, people first met serotonin, it was part of like what happens when you get cut. People were actually, no way, it's not in the brain. It's like, but it's important in the brain. It's also a major neurotransmitter in your intestinal tract. We're going to talk about the enteric nervous system. So serotonin plays many different roles, but it's an important neurotransmitter in the brain. I should also mention while we're here that 5-HT, 5-hydroxytryptamine, that is just another name for serotonin. Serotonin can be serotonin, serotonin, or 5-HT is just the way people in the business usually write it, as just a shorthand. It means 5-hydroxytryptamine. That's just its chemical name. And here, he's saying, there are lots of different types of serotonin receptors. The 5-HT1A, 5-HT1B, blah, 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 5-HT2A, 2B, 2C, la, 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 la. So this is typical. There's usually many different subtypes of receptors for any particular endogenous ligand. When we get into acetylcholine, you've already met the nicotinic cholinergic receptor, and we're also going to meet the muscarinic cholinergic receptor, and we're going to meet a whole bunch of adrenergic receptors, and there's lots of different versions. Sometimes they're localized in different parts of the body. Sometimes they have different specific kinds of roles they play. So let me mute this person again there. So it's very typical to have more than one subtype. This particular presentation here is focusing on this particular subtype of serotonin receptor, the 5-HT2A receptor. This one has garnered particular interest because it seems to be one of the common points of all the psychedelic drugs. If you look at different psychedelic drugs, most of them bind a number of receptors. You take something like LSD. LSD is something that they usually call a very promiscuous ligand. It likes to bind 5-HT2A, it likes to bind dopamine receptors, it likes to bind all sorts of receptors, right? So just because you have an exogenous ligand that binds a receptor, it might actually bind more than one type of receptor and different receptors with greater or lesser affinity, right? So given that a lot of these psychedelics are promiscuous, meaning they are going to bind more than one kind of receptor, when they're trying to figure out, well, what's the common theme here? This 5-HT2A receptor seems to be kind of, no matter what other things they're binding, they all seem to get a piece of this 5-HT2A receptor. So it's been something that people have looked at with particular interest. And so this whole presentation is just focusing on the details of that particular subtype of receptor. That being said, everything we're going to talk about in terms of what does it mean to bind a receptor and all that, that's more generic. It's the basic idea of what binding is and how it creates the activation of a G protein is kind of the general story. Okay. Let's see. It's not unusual. Most receptors have at least 2 or more subtypes. So now, in order to really understand how this works, you have to really think about it at the level of the atoms and the molecules and the chemical bonds. Again, which is why we've been beating this into the ground, is like something, it's like, but I don't want to learn the chemistry. I just want to know, like, why do you trip if you drop acid? You got to know it. You got to understand what a polar or a non-polar molecule is before you can go there. So non-polar interactions, we all know that, right? That's just when you have things with no charge imbalance. So things tend to come together that tend to be non-polar. They tend to get kind of squeezed together. So things that are non-polar tend to like to nestle in with each other. Hydrogen bonding. You have seen this a bunch. Local charge imbalances. It's what keeps water molecules together. It's how ammonia here is being connected to water or whatever, to itself, right? So hydrogen bonding, local charge imbalances that are going to attract things with these electrostatic attractions. So that's classic. Ionic bonds, where you totally give up an electron, something totally takes an electron, and now you have a positive and a negative that then are attracted. And we saw it in the context of like sodium chloride, where an entire atom gave up and another entire atom accepted. But here we have, you know, this little like ammonia thing giving up an electron, and this taking an electron. You can make ionic bonds between different parts of a more complicated molecule like this as well. So it's not necessarily just like two atoms that, hand off an electron. Here we have two molecules, more complicated molecules that have handed off an electron and now are attracted with an ionic bond. So they're saying serotonin is a perfect example here. And the idea here now is when you look at a molecule like this, there's two things that are important. Whenever we want to think about a ligand, the two things we think about, one is there's the structure of it, the physical shape, because that physical shape is going to have to fit into, basically the binding site, which is going to have a particular, it's kind of like the shape of the binding site and the shape of the ligand, they have to physically fit. You can't stick a square peg in a round hole. But then in addition to the physical structure, you also have to think about the chemical bonding. Right, so if you can't fit a square peg in a round hole, but even if you had a round peg, if the round peg was positive and the hole was positive, it still wouldn't go in because it's getting repelled by the charge. So you need to have both the shape has to be right, but then the kind of bonding that the different parts of the shape make has to be correct as well in order to really fit, to have the key fit the lock, so to speak. So if we look at serotonin, stuff we've been talking about, hydroxyls, what kind of bonds do hydroxyls like to make?
Ionic bonds.
No.
Hydrogen bonds?
They make hydrogen bonds. So they don't actually give up or accept an electron to make a, that more extreme ionic bond. Instead, there's a local charge imbalance, and they're going to make hydrogen bonds. This nitrogen part of the ring here, this also likes to make hydrogen bonds. This part, the amino group here, this part actually does like to make ionic bonds. This one will give up an electron, which can be accepted by something else to make an ionic bond. So we have parts that make hydrogen bonds. We have a part over here that does make ionic bonds. There's parts of this ring here where there's relatively little charge imbalance, where it would be more of a non-polar kind of area. So different parts of this thing like to participate in different kinds of chemical bonding. Hydrogen bonding, ionic bonding, non-polar bonding, or interactioning. So that's important. The structure, the shape of it, but then the different parts of this shape like to participate in different kinds of bonding interactions. So hydroxyl makes hydrogen bonds. The ring is nonpolar. Amino group can make ionic bonds. The ring nitrogen can form a hydrogen bond. So kind of looking at this thing now, rather than just like a ligand, you can think of it a little more functionally now. Like, you know, a key is not just a random key. A key has to have just the right little grooves cut in it. Otherwise, it's not going to work, right? So this is kind of thinking about it that way. The reason why this is going to bind the receptor is it's got just the right shape and the right properties to fit into the lock. So that is the ligand. We've talked about the ligand. The other part of this is we need to now dive down into the lock. So this is the key. The serotonin is the key. Now we need to see the lock. What are the properties of the lock that are going to fit the key? And to do that, we need to go into protein chemistry, right? Because the receptor is a membrane protein. So here we go. Boom. Again, amino acids with its amino group and the carboxyl group and the side chain, that RR group. And remember, there are 20 naturally occurring amino acids. Each one has a different kind of side chain. Some of these like to make hydrogen bonds. Some like to make ionic bonds. Some are nonpolar. It's going to be important in a few moments. You know, you take peptide bonds and you assemble up these, and they, what do we call the just order of amino acids you're connecting up? What's that structure called?
Primary structure?
That's primary structure. And then if we start going into helices, what do we call that?
Secondary.
Yeah, we start getting into secondary structure, but let's start with primary structure here. So this is an important point here. If we've thought about different amino acids being different, there's valine and leucine and this and that. And so he says, like, is it kind of like a string of beads? You know, it's like, oh, it's different amino acids hooked up in different orders. And he's like, no, not really, because it's not like different colors. These different side chains are pretty different from each other. So he's like, oh, so more like kind of a charm bracelet. And he's like, that's still not a good analogy, because still the charm bracelets, the charms are kind of just for show. And the side chains on actual amino acids they have very functional functional activities. So he's saying the best way to think about a protein is like a tool belt. You know, this idea, if you have a tool belt, you have particular parts of this belt that are specifically optimized for certain kind of interactions. You know, the little hammer clip fits a hammer really well. If you try to put the hammer in the pocket that fits the tape measure, it's not going to fit. It's not going to work. If you try to take the tape measure and put it in the thing that holds the hammer, it's going to fall out. It's not going to stay there. So this idea of a protein and the different amino acids with their different side chains, you really want to keep in mind that different side chains make very specific kinds of interactions. And there are certain things it's going to want to interact with and hold on to, and other things it's really not going to interact with or hold or bind to. So does that make sense? So that's an important thing. When we talk about how the serotonin molecule binds to the receptor, you really want to think about those different aspects of the serotonin molecule we talked about, having these particular kinds of interactions with different side chains on the amino acids of the protein that makes up the receptor. So let's continue here. So iPhone, for whoever is whose iPhone, please mute. You keep kind of coming back in. I have to keep remuting you. All right, nonpolar side chains, polar hydrogen bonders, polar negative ionic bonders, polar positive ionic bonders. So these are all these different different amino acids, and kind of giving you a sense of what kind of interactions they like to do. You know, this is just, again, you don't need to remember this, but I want you to you should know the big picture, that some of them like to do one kind of bond versus some like to do another kind of bond. I don't care if you, I could not, if you said, asparagine, I couldn't tell you like exactly what it does if I don't look at the chart. So don't worry about memorizing, but do know the idea that they are the ones that do to these different bonds. Then it's important to think about the three-dimensional structure of the protein, because that's going to be important for this. And again, in this picture here, we have these are these alpha helices. Each of the colors is representing a different amino acid, like amino acid, amino acid, amino acid. Every third amino acid is making hydrogen bonds to become stabilized as an alpha helix. In my membrane, this is going to be the bilayer of my membrane. This is going to be the outside of my cell.
Oh, Professor, you froze. Well, on.
All right, so it's showing up again?
Well, in my computer, you're a little froze. Let me check. Maybe it's just me.
Yeah, I think it's Maria's connection.
Okay, thank you, guys.
Although it's frozen on, here, it's going really slow. I have it like coming out on my iPad. It's coming out really slow. I don't know why. All right, so we are going to have the membrane protein that is the receptor looking kind of like this. There's going to be 7 transmembrane helices that go down and up and down and up and down and up. There are going to be loops that are on the outside. That's what the receptor presents to the ligand. When the serotonin comes in here and drifts in, it's going to be on this side. On the cytoplasm side, this is where I'm going to have my G protein hanging out. So this is kind of the basic layout. We're going to have this protein that has 7 transmembrane helices. And this is a classic. This 5-HT2A receptor is one of these 7 transmembrane helices, but a lot of the G protein coupled receptors have the same basic structure. So again, remembering an alpha helix is just this idea that you have every third amino acid is going to hydrogen bond to the one and stabilize in these helices. Again, you've met this already when we just talked about secondary structure of proteins. And then the idea that a receptor of this type is basically 7 transmembrane helices going up and down. And these exoloops, these are the parts that are visible to the outside world. The cytoloops, those are the parts that are visible to the inside of the cell, the cytoplasmic side. So does that make sense?
One question, Professor. The G protein binds to a cytoloop?
Yeah, so so the G protein is going to be ultimately down here.
Okay, thank you.
And basically what's going to happen, or I should say right here, here's the G protein. Basically, when the ligand binds up here, it's going to tweak the orientation of these helices, change the shape down here, and that is going to activate the G protein and turn it on and start this whole process. right? So by having the ligand bind up here, it's going to push and pull on these helices, which changes the shape down here, which is going to activate the G protein. Okay.
Is the ligand always binding on the N-terminal side?
On this exoloop side, just, yeah, so think about the just think about it more as the parts that are visible to the outside of the cell. So it is, yeah, so this, I mean, this, yeah, so that's clear. So now, this is kind of showing it a little more clearly as well. You've got the receptor, you have the cytoloops, which are the side here that are connected to the G protein that are going to activate. You have the outside, which is the part that's the where the ligand is going to actually come in and bind. And again, when the ligand binds the receptor, that is going to tweak the orientation of these helices, which is going to activate the G protein and start the whole process we talked about. So now, this next part, again, don't worry about the details. This is, I'm doing this just to kind of give you a realistic sense of what does it mean when you have a serotonin molecule come in and bind a serotonin receptor. You are not responsible for any of the details I'm about to say right now. Just so you don't, you can just watch, don't take notes about it. So the basic idea, we are view from above. We have the serotonin molecule is drifting in. It sees this view of the cell membrane here. There's the serotonin molecule drifting down. The green is supposed to be all the transmembrane helices of the protein, that's the receptor. So now we're coming in to bind. So does everybody, this is kind of point of view, looking outside the cell, looking at the serotonin molecule, drifting down, about to get into the pocket, into the receptor. And again, remembering that there are these receptor attachment sites for the serotonin, parts that like to make hydrogen bonds, parts that make nonpolar bonds, parts that make ionic bonds. Wait, I need to clear. How do I clear all that? Oh, well. So on the third transmembrane helix, on the third transmembrane helix, the amine group of serotonin forms a strong ionic bond with an aspartic acid and also a hydrogen bond with an adjacent serine. Right, so basically, here's the amine group, and it's going to form an ionic bond here, and it's going to make a hydrogen bond with that dude up there. Right, so we have two different things here. We have a pure ionic bond, but since this is charge imbalance, it's also making an a hydrogen bond there. So that's part of, it's got the right, these pieces of the molecule are in the right physical place, and they are making the right kind of bond. They have the right kind of charge imbalance to actually grab and hold in to the side chains of the amino acids that happen to be there. You know, on transmembrane helix 5, a serine forms a hydrogen bond with the nitrogen of the indole ring. So here's the nitrogen of my little indole ring on the serotonin. Here is this serine, and they are making a hydrogen bond. They're holding together here. And again, meanwhile, we still have my ionic bond here and my hydrogen bond here with the amine group. So we can see this thing really starting to lock into place. The physical shape is important. These different parts of the serotonin have to be in the right physical space. And again, they're making the right kinds of chemical bonds that are just the perfect complement to what the side chains of the amino acids that are found there want to make. Right, so this is making sense? You know, on transmembrane helix 6, 2 phenylalanines and a tryptophan contribute to a local hydrophobic pocket, which attracts the nonpolar indole ring. You know, so this indole, you know, this nonpolar here likes to fit into the little pocket that's nonpolar. You know, and meanwhile, the hydrogen and ionic bonding and hydrogen bonding, that's all still happening. You know, so all of these interactions are all happening, changes the position and conformation of several transmembrane helices. So as all of this stuff is happening between the serotonin and the receptor, it actually tweaks the orientation of those transmembrane helices. Right, which is what I was talking about. So when those transmembrane helices move, that changes the shape of the cytoplasmic side, and that's what's going to activate the G protein and start that whole process we were talking about. Right, because the shape is necessary for catalyzation. Things catalyze when the shapes fit just right. So by having the, you know, this should say serotonin, by having the serotonin bind at the receptor, it tweaks the helices just right to change the shape here, which activates the G protein. The G protein is now activated, turns on adenylyl cyclase. Adenylyl cyclase starts making cyclic AMP, and we're kind of good to go. So does that make sense? So just to give you an idea of what does it really mean. And now we can show, like, why is this, how does this like relate to exogenous ligands? So I'm going to, the last part of this presentation here, let's look at psilocin. Psilocin is basically like if you eat magic mushrooms, it's psilocybin, but as soon as it gets into your digestive tract, it gets transformed into psilocin. So this is what is in your body if you've just eaten some magic mushrooms, psychedelic mushrooms. And if you look at this molecule, it's awfully similar to serotonin. It's a little different, like the hydroxyl is moved over, you know, one place on the ring. There's methyl groups here. Instead of just having hydrogens on this amino group here, we actually have a methyl group, like adding in an extra carbon and hydrogen. So this is a little more kind of developed out. But for the big picture, if you look at them, they're awfully similar. It is not going to be shocking that that psilocin molecule can get in and have some effect at the serotonin receptor, because it has pretty much the same shape. In general, those different areas tend to make similar kinds of chemical bonds. So people have like modeled what happens if you take this and put it in. And basically, it's what you think happens. It gets in there and it does bind, but it doesn't bind exactly the same way. You know, it's basically saying, you know, that due to the large methyl groups on the amine, you know, the you don't have the hydrogen bond, you know, with the amino group that you would with the with just serotonin. So it's not exactly the same. You know, he's basically talking about basically all this stuff happens. The G protein can bind, but the slight alteration in the configuration of the docking site causes changes in function. Instead of the full agonist effects of serotonin, psilocin acts as a partial agonist, altering the activation state of the G protein population. So it actually will turn on the G proteins, but not as fully as if you were going on a full serotonin. So it turns it on, but it's a partial agonist, which we didn't even talk about. That's another option. But this is why there is an effect, in your consciousness if you eat magic mushrooms, is those psilocin molecules are flooding around your neurons, and they have a shape that is really close to the serotonin. They get into the serotonin receptors, they activate them, not fully, but they do activate them nonetheless. Plus they're activating them all over rather than just at specific synapses, and that's going to cause some major changes in your kind of consciousness and sensory perception and all that. So, so other questions about this? I don't want you to remember the details about how serotonin binds to a receptor, but I do want you to remember the basic ideas, that the shape was really important, that the specific kinds of chemical bonds that it made at those different regions of that shape is really important, that that's what's going to create the efficaciousness of what is going to make something an endogenous ligand. It means that this molecule has a shape and chemical properties that can mimic the endogenous ligand, kind of the same way psilocin mimics. It's not exactly the same, but it's close enough that it fools the receptor and gets in there and binds and makes something happen.
One question, Professor. Are ligands involved in the feedback loops? For example, if you have the magic mushroom and that attached to a receptor of serotonin, that can you produce less serotonin because it's attached, or I'm going
no, so
another way?
There are, in fact, so there are a lot of feedback loops like that.
Okay.
Part of the way that like the SSRIs, like Prozac, a lot of those antidepressants work.
Okay.
They go and they increase the amount of serotonin in the synapses of your neurons, and that actually causes a down regulation of the serotonin receptors on the neurons. So it's
okay.
like kind of if the volume is always really loud, you're just going to always walk around with earplugs in or something. So like if you're taking Prozac and there's always a lot more serotonin around, then the cells tend to just lose their sensitivity to serotonin.
Okay.
Or with the same thing with opiates. Like if you take morphine, like 30 milligrams of morphine, the first time you take it will be a heavy dose and will definitely do the job. But if you've been taking it every day for a week, you might not even feel that at all. You might have to take, twice that or more to get a good effect because of the same basic idea. You start down regulating the receptors. You basically kind of reduce sensitivity. I was talking to somebody earlier about that same thing, like the pills that you can buy as a prescription for morphine for somebody that is on maintenance pain meds are really high dosages. You can buy dosages, you can get a pill of morphine that if a drug naive person took, they would die. But somebody who's taking morphine every day, it's the amount they need to get get pain relief because of that same thing, that kind of negative, that feedback loop you get. If you have high levels of the ligand around a lot, the cells tend to downregulate the number of receptors to that ligand, so kind of to reduce sensitivity to it.
So that's like your tolerance going up.
Exactly, that's tolerance.
Is that reversible?
Yeah, if you just if you just wait a little while, it goes away. like if you took LSD at 100 mics one day, it would be a strong trip. If you took 100 mics the next day, you might barely feel it. But if you took it like a couple of weeks later, it would be the same again. You know, so basically, whenever you put drugs in your body, you know, things dynamically shift, and when you take a holiday from them, things kind of shift back. So,
Thank you.
sure. All right, so we are, wow, it's 10:04. Let's come back at 10:40 to give you guys a break. That's been a long road there. So let's come back at 10:40, and we are going to kind of talk about science and literature and all of that. So I'll see you all at 10:40.